Thank you so much to the heart failure panel. And we have our first cross-AMP discussion panel coming up onto the stage next. The timing of this panel was very deliberate in that we wanted to have the 4 programs that presented yesterday, as well as the heart failure program that you just heard from, who all have data portals with these very rich multi-omics data sets that are all sitting in isolated spaces in their own portal space. And so we have a session that's moderated by Susan Gregorick, who's the director of the NIH Office of Data Science and Strategy, to discuss how we can get data interoperability across all of these different AMP portals and how that will increase the value of the data sets.
Welcome. Wow, it's such a fantastic meeting. I've learned a lot about all the different AMPs. I'm just so glad that I've been able to attend. I'm truly impressed with the accomplishments from the scientific achievements and the scale of the data that's generated within the AMPs and the publications that have resulted from the work that you have done. It's such an exciting time. I can imagine how the larger community can leverage these data to further improve our understandings of disease pathways and to enable rapid selection of targets for treatments. Having the ability to use transcriptomics, metabolomics, phenotyping, imaging, genomics from tissues collected across the AMPs like AMP-AD with similar data sets collected from AMP-CMD and AMP-AIM and AMP-HEART to reveal common molecular pathways from across our AMP systems, and then to identify abnormal perturbations based on comorbidities. That would be really exciting. And it would really move us beyond a taxonomy approach, but to a approach that's really at a mechanistic level. So what we want to do this morning is to hear from our data experts on our panel about their portals, about what's possible today, about what they would envision for the future if we really were able to accomplish that goal of interoperability and data integration. And then what would success look like if we were able to do that? In their view, what would we accomplish? What kinds of science would we enable ourselves to do? So with that, I'm going to turn to the first slide. Do I do that? Maybe I do that. Yes. To introduce our first speaker, and then I'll subsequently introduce each speaker sitting there. But it's really a pleasure to introduce my friend and colleague, Dr. Zuzana Pantachevska. She is the Director of Strategic Development and Partnerships in the Division of Neuroscience at the National Institute of Aging. You've heard her speak earlier. Her program focuses on developing systems biology and systems pharmacology capabilities for AMP and for Alzheimer's disease research and drug development with an open science framework in mind. And she serves as the NIH co-chair for AMP-AD 2.0 and in the development of the newest program, the AMP SBI Systems Biology. And with that, Susanna, thank you so much. It's a delight to have you here.
Susan, good morning, everyone. It's so wonderful to see the confidence monitor on. And I brought my glasses just in case. So what a day has been yesterday and this morning. Many discussions last night among the different teams. And as all that transpired, I was reminded by something that Dalai Lama, or maybe it was somebody on Facebook, said. AMP, you're perfect, and so much room for improvement. So I wanted to, all right, that's me. So my comments are really going to focus on the translational impact of the open science and fair data practices that we brought in very deliberately into the AMP-AD program 10 years ago. So you saw Anna present this yesterday, but from the get-go, we all want, of course, to accelerate discoveries and make discoveries translatable. And so we thought that the secret ingredient would be to focus on open science practices and fairness of data. And so we designed from the get-go not only the funding incentives to align with these goals, but also for the enabling infrastructure to support these goals. So this was, again, you saw this, the commitment to fair data, streamlined governance, rapid sharing of data, distributing these assets not just to the AMP-AD partners, but to the greater community at large at the same time, no publication embargo, and focusing on being user-centered. Also, a really serious effort and commitment to enable diverse set of users in various ways. So what that has helped the National Institute on Aging is that over the past 10 years to launch several follow-on programs that were really enabled truly by the output of the AMP-AD program. So there are two translational centers, the Model AD Center, whose mission is to develop new mouse models based on the complex genetics of AD, and modeling various features of the late onset sporadic disease. And these centers are using the AMP-AD data and network modules to establish this molecular homology between human and the mouse models. And so these also operate as open science programs. Then several years later, we launched the TREAT-AD translational centers that are using that massive output of data and target nominations from the AMP-AD program and prioritizing a subset of targets for the development of target-enabling tools that are then making it possible for biologists as well as drug discoverers to more deeply study the disease biology of these pathways lit up by the targets, or to advance a set of novel targets into drug discovery. Another open science enterprise. So here is the Model AD website. These are two large translational centers. And again, they are developing mouse models as precision medicine research tools. That the development of these models is heavily rooted in the human data. And so the AMP-AD output comes to play, you can see a series of these new mouse models and how they align, how the transcriptional signatures of the mouse tissue, brain tissue, aligns with the human transcriptomic modules that were developed by the AMP-AD program. So that allows us to establish how relevant the model is and for which aspect of the disease biology it is most useful for. And so the Model AD also has established a very rigorous preclinical testing pipeline, and that allows promising preclinical drug candidates to be matched with the most appropriate mouse model that reflects the biology of the disease, and then to develop translational phenotypes that can track the efficacy and translatability of that compound. So we also, because this is an open science enterprise, we distribute the mouse models and all the data to everyone, no strings attached. So here you can see the uptake of these mouse models in the academic and industry communities. And there is a comprehensive list, and all these models are available through the JAX repository for purchase. So the other translational center is the TREAT-AD Center, and you heard yesterday that we have a really robust output. We have over 1,000 unique candidate targets coming from unbiased systems biology analysis. So you can't, of course, take 1,000. They're not all tractable. How do you prioritize these and winnow the list? So the TREAT-AD centers are missioned with that. They're taking the output of AMP-AD and other target discovery programs and developing open source research tools all the way to chemical probes on one end and preclinical drug candidates on the other end, and distributing all this knowledge and tools freely, openly to all. And so here are what are these assets. Some of these assets are data packages for those prioritized targets, but then there's this physical matter, everything from knockout cell lines, validated antibodies, crystal structures, and all the information on these, both informatics packages as well as the physical material, can be found in the knowledge portal and the Agora platform. And here again, this is the TEP Target Enabling Packages portfolio that you can find on the dashboard of the TREAT-AD centers. And again, it is all made available to both academia and industry communities. And the last translational program that we were able to launch and build out in a very robust way is our combination, drug repurposing and combination therapy program. Here we built a program over the last 4 years that brings together, we now have funded over 40 projects. These are highly cross-disciplinary programs where the teams are leveraging not the AMPD data, but not only the AMPD data, all kinds of publicly available data, all the way through claims data, EHR data, and making predictions on what could be the valuable repurposable drug candidates. We have identified several dozens of these, and even taken some of these to phase 1 and phase 2 clinical trial for AD. What we're currently doing, just like we aggregated all the target data from AMPD and Agora, we're now aggregating all these repurposable drug predictions so that we can have a data-driven process of prioritization. So we continue to build in this open science, an open science research ecosystem. So all these programs, starting from AMPD, are contributing through the centralized infrastructure, as well as taking data and making into the next step of translation or broadening or deepening the discovery. So the portal, again, infrastructure is constantly evolving. It was enriched recently with the Model AD Explorer, where biologists, clinicians can, in an easy and visually intuitive way, explore the new mouse models and how they are aligned to which features of Alzheimer's, from the molecular to pathologic to biochemical. And you heard from Anna, we are continuing to build on this. This is an infinite game. This is not easy. But imagine a world where we can really explore the causal, shared and distinct causal biology across chronic disorders. So we heard the very moving presentation by Cynthia earlier today. Imagine if we're able to treat the causal biology, particularly for elderly patients with a lot of comorbidities, and not just treating symptoms that, while we treat one symptom, we actually introduce another risk factor or another disease condition. So we are humbled and inspired, and I look forward to doing more and better. Thank you.
Thank you so much. Our next speaker is Dr. Noelle Burt. She's the Director of Development of Operations for Diabetes Research and the Knowledge Portals at the Broad Institute in Harvard and MIT. She's a human geneticist by training. Her work focuses on the design and development of open access resources for biomedical research in the community, and she's the Director of Operations for AMP T2D, Data Coordinating Center. And so with that, Noelle, thank you so much.
Thank you. I'm grateful to be part of this panel today to talk about interoperability and integration across the AMPs, and to represent the AMP CMD Knowledge Portal and discuss its approach and impact over the last 10 years or so. Before AMP T2D even existed, we were very lucky to be funded as the Data Coordinating Center for the International Type 2 Diabetes Genetics Community. What that meant was we were both generating and aggregating all the individual level data for genotype level data across a set of cohorts for type 2 diabetes and related traits. What this meant was we were responsible for QCing, harmonizing, and distributing amongst our colleagues as human geneticists all this data. And what this meant was we had a really great community in the type 2 diabetes space to share data and build trust. Along comes AMP T2D, enables us an opportunity to actually make this data more useful by the broader community. So in 2015, we launched the first portal for type 2 diabetes, which was only with 9 data sets and 25 traits. But what was very unique about this at the time, it was the first portal of its existence to share full summary level statistics, open access to the research community and to you all, who can hopefully use them in target discovery and identification, first of its kind. And so what was wonderful about this is that it led to the Type 2 Diabetes Knowledge Portal, which was matured over time and really has as its core essence making genetic data accessible to others, right, in a useful framework. So if you're not an expert, you can use this data. And so this has evolved over the years and has led to a publication that just happened in 2023. But really, what was really great about this and what the platform for AMP T2D allowed us to do was to help other communities, other genetics communities were struggling with the exact same problem at the time. They had a lot of data. They wanted to have it aggregated, shared, stored in a consistent place, but they wanted to make it accessible to others. And we were very lucky that this was done across the AMP CMD disease indications, starting with cardiovascular disease and cerebral vascular disease. But what was great is we were able to bring those community data sets and community experts to the table to help contribute to this broader resource. So this was all open source and open access at the time. So this is AMP CMD Knowledge Portal existed before the actual program existed, because it was these portals that had come together with all this data contributed by the community. And what this meant was we were to take the same exact approach that we took for the Diabetes Genetics Data Coordinating Center and work with all of these consortia to actually have a partnership to steward and represent their data as they wanted it represented. We also had curation and research to actually aggregate data from open source resources and broad resources like the GWAS catalog. And what this has led to is a resource that has over almost 600 data types across almost 700 phenotypes, but with direct contribution from contributors. This means they talk to us, we talk to them, and we do it exactly as they want. This is outside our consortium, which has been really cool, because we as an AMP platform have ingratiated the trust of broader communities that have their own consortia, right? So what this meant was, geneticists have done a really great job of identifying causal variants. And we're assuming, based on statistical prediction, that we have organismal function licked, right? But there's this whole space in between. This is all a biological model. So all of this work in between still had to be done. So enter AMP CMD, allowing us to really think about how to fill that gap in the middle, generate new data, aggregate that new data, enter Kyle Galton and the Common Metabolic Genome Atlas, which allows us to bring in individual raw data for gene expression and other genomic data to help us adjust the space in between. What are the tissues, cell types, mechanism of action for all these variants we've identified over the years? So what Kyle did, as we talked about yesterday a little bit, was he built a technical replicate of essentially the ENCODE model to really bring together gene expression data and other regulatory raw data relative to the CMD tissues and aggregate that in a platform that is open access. But what's cool about this is he took the exact same approach that we took for the genetic data and worked with disease-agnostic resources, GTEx, ENCODE, Blueprint, Chip Atlas, HGA, integrated those, but also went after other like-minded resources who have disease-specific genomic data like HPAP and KPMP. These are all integrated and protected and stored in an ENCODE-specific model that Kyle mentioned yesterday. But these are all available on the Knowledge Portal. And how do we make that available? Well, the challenge here is now you have the substrate, right? You have genetic data and you have genomic data. But to really integrate these data and get hypotheses about the molecular and cellular function, you have to run a suite of tools. A lot of methods that, again, are community contributed. These are the best practice methods. So we implement these in software. These include meta-analysis, gene-to-variant predictions, tissue enrichment, genetic correlations, and all of these results are accessible to you on the Knowledge Portal. So what are the impacts and the products that really come from this effort? Well, it's a flexible software platform, which we like to call Huge AMP, or the Human Genetics Amplifier, which is a federated resource, allows us to store and process individual level data, generate summary level statistics, make those accessible. It's federated both with a European node and with the CMD Genome Atlas as well. This is fully open source. So this is one of the big outputs of the program. Additionally, I think the best part is it changes the way that translational biologists or folks in industry can interact with these resources. This is what it used to be like. You'd have to go to all these resources, you'd have to be pretty substantially computationally savvy, integrate all this, and then make your favorite choices about which gene to pursue for target validation. Well, now it's one-stop shopping. You go straight to a resource, and it's presented for you in a way that is community-driven, open access, and hopefully very clear. So in addition to that, we also have novel approaches that we've run with our community experts, bringing us results and methods that they want us to encode. We make those accessible, and what Melissa even showed yesterday is a very simple UI and a simple framework for you to interact with. And these allow for novel approaches for basically human genetic support, right, for your favorite gene or target. Beyond that, we also still try to ingratiate the community's contribution by working with them to contribute their curated effector gene results. So a lot of these people are doing this work on their own, where they are layering genetic, perturbational, regulatory evidence and creating their own curated list with all these evidence sources. We've encoded and brought all of those into the Knowledge Portal so that you can view across over 150 curated effector gene predictions in the context of all the automated results that we've produced. So the resource over time has been pretty awesome to be part of, and this is just to give you a sense of the growth, but right now we have about 8,000 users per month, and we're really excited to think about how we take our unique approach and marry it with the best of these amps that you're going to hear more about today. And so one example of that is that we were very lucky to work with the NIH to be funded as a community genomic resource. So take this exact huge AMP platform that we've done for common metabolic disease and apply it to common disease. And what's really great about that is some of the AMPs that are represented here we've started to work with to integrate their genomic and genetic data to do the exact same thing we've done for the AMP CMD knowledge portal, but do it in an open framework for a community genomic resource. So this is just to end with our philosophy and approach, which I think will be useful as we talk later. We really work beyond our consortium of the AMP program as well to collaborate directly with investigators who have expertise in our disease indications and the genetics and genomics. We onboard pre-computed results, and we also encode any type of analytical approach that our collaborators think is very important and make those accessible, right? We incorporate external resources, whether they be disease agnostic or disease specific. And finally, hopefully, we integrate them a platform that's accessible via API or visualizations to make your life a little easier. And with that, I'll end, a little bit ahead, I don't know how that happened, with this team of lovely people I get to work with. And one thing, given being part of an AMP for 10 years, there are a lot of people that are not represented on the slide who have gone to bigger and better things, and it's really been wonderful to be part of this endeavor. So I'm looking forward to the rest of the panel. Thank you.
Thank you so much, Noelle. This is great. Our next speaker is Sweta Ladwa. She serves as the Chief of Scientific Solutions and Delivery Branch in the Information Technology and Applications Center within the Office of Management at NHLBI. She supports NHLBI's data science initiative and leads data management for NHLBI's cloud-based data science ecosystem, Biodata Catalyst, as the Scientific Program Director for the Data Management Core, while also working across NIH, and I work with Sweta on many programs, to help support our initiatives in fair and interoperable data. Sweta, thank you so much.
Thank you very much. It's a pleasure and honor to be here today to celebrate all the progress that's been made over the 10 years of AMP. So we thankfully heard a lot from the heart failure team this morning, and they did a beautiful job of describing the relationship of the work they're doing with Biodata Catalyst. We might be the new kid on the block, but we do bring a lot to the table. And Sanjeev mentioned that the heart failure AMP is leveraging NHLBI's investments in the HeartShare program. And they're looking to bring together multimodal data from large-scale cardiovascular disease epidemiological studies, multi-center heart failure clinical trials, and other studies to understand HFpEF, as well as develop algorithms to further explore the subtypes for HFpEF. And so for the heart failure AMP, there was not a need to create a new portal. What was done was the investments that NHLBI has made over several years in the Biodata Catalyst platform was leveraged to support the heart failure AMP, because Biodata Catalyst has a lot of capabilities and functionalities and platforms that many have heard about over the past 2 days. And so we've been leveraging that sustainable resource, and as Susan mentioned, we've been working with several trans-NIH efforts to leverage that same sustainable ecosystem for programs such as the Recover program for COVID-19, as well as the Regenerative Medicine Innovation Program, both of which are congressionally backed programs. So in terms of Biodata Catalyst's mission, it's to develop and integrate advanced cyber infrastructure, leading edge tools, and fair data to support the NHLBI research community. And the vision is to be a community-driven ecosystem implementing data science solutions to democratize both data and computational access to advance heart, lung, blood, and sleep science. Biodata Catalyst is an ecosystem, and for others that may not be familiar, we are multi-platform. So what exists under this cloud-based ecosystem is multiple platforms that support different functionality, including analytic workspaces, cohort building tools. There's authentication and authorization built in, because we do have individual level data that we need to make sure is governed appropriately in line with the consents of the individuals who participated in the various studies. But we do house both the data and the computational resources under one roof. It's a one-stop shop. And we're here to support our researchers to accomplish what to drive towards discovery. We are multimodal in that we support various multi-omics data as well as clinical data, imaging data, various formats of sleep data, such as actigraphy, polysomnography, ECGs. So there's a lot of data. We have over 4 petabytes of data currently in the ecosystem, and that's not including all the imaging data, the multimodal imaging data that the Heart Failure AMP is bringing into the ecosystem as part of their efforts. The ecosystem is free to access. There are computational and storage costs that may be associated with the compute, but it's at cost. It's at a discounted rate thanks to the NIH STRIDES program. But under the hood, we do have several platforms, as I mentioned. 7 Bridges, which also supports the Cavatica platform, as we heard about yesterday, provides analytic capabilities, including machine learning and other capabilities. We also have the Broad's Terra platform in Biodata Catalyst within the ecosystem, and Terra supports a couple of the AMP programs as well. We have teams that I mentioned support the authorization and authentication layer, which is at the UChicago, it's the Gen 3 team. Harvard supports phenotype-based cohort search across all the different data that's in the ecosystem, and that's from the Picture tool. We have a team at UNC Renzi that supports the coordination of all these efforts, and we also host the TopMed imputation server within the ecosystem that was mentioned yesterday as part of the AMP-PD GP2 project. So there's a wealth of data available and a wealth of analytic capabilities available under the ecosystem to support collaboration and sharing and fair data science. So I mentioned some of these. We do have 4 petabytes of data, including the most ethnically diverse set of whole genome sequences from TopMed. Extensive longitudinal cohort data from multiple ethnic groups across the United States, clinical trial data, sleep data, and data from the COVID-19 pandemic, and we're bringing in imaging data very soon. We have over 200 whole genome sequences from the TopMed data in the ecosystem today, and that's continuously growing as the TopMed program is expanding, as well as the multi-omics that they're generating as they move forward in their efforts. We do also have data from the active trials that was mentioned yesterday, some of the active trials, not all of them. But we do have data from over 75 studies that NHLBI has invested in longitudinally over generations and decades, and over 350,000 individuals represented, and that is continuously growing, especially as a result of the NIH data management and sharing policy that Biodata Catalyst also is supporting investigators in achieving compliance with. I went over this briefly, but we are providing an infrastructure that allows researchers to bring their own tools and workflows to the data, as well as use the published tools within the ecosystem. They can collaborate on with multimodal data and multimodal analytic capabilities in the workspaces. We have Jupyter notebooks, R, Python, SAS, as well as capabilities to facilitate GWAS analysis, variant calling, image visualization, machine learning, as well as app development and hosting, pipeline development in multiple workflow languages, and search at various levels. So Biodata Catalyst is working towards interoperability in light of NIH's mission to support a federated data ecosystem. We are working at the NIH level with the Cloud Platform Interoperability Effort, NCPI, to federate data analysis, to bring the compute to the data wherever it lives in the NIH enterprise. And that also includes support for the NIH Researcher Auth service that helps authorization to access data follow researchers wherever they may decide to compute on that data in the NIH enterprise. That also includes support for FHIR for EHR data, and we are establishing FHIR servers to be able to exchange EHR data, as well as supplement that EHR data with distinguishing social determinants of health data that's coming from the EHRs, as well as our cohorts. We're also working with the Advanced Research Projects Agency for Health, or ARPA-H, in the Biomedical Data Fabric Toolbox to support a national unified consistent layer of data services to work across many different systems and environments, as well as working globally with the Global Alliance for Genomics and Health to work to interoperate with international data sources, including the UK Biobank and the NIH All of Us Research Program. We're really working to establish a reusable and interoperable and sustainable product to make research possible. And we're looking to leverage advances in technology to enhance the infrastructure so that researchers can focus on the science and not have to worry about all the things that they need to do to get there. And so that includes looking at generative AI technologies, different ways to incorporate large language models or homomorphic encryption, looking at ways to establish ephemeral workspaces and the like. But data is going to be flowing because of the NIH data management and sharing policy, and there is a need to establish good interoperable infrastructure to support that secondary data research reuse to drive towards discovery. Specifically for the AMP, we've worked very closely with the teams to establish image de-identification protocols for imaging that's critical to the heart failure program, but that will be extensible beyond that, and especially with regards to all imaging modalities, including the echoes, the ECGs, MRIs we heard about earlier today, as well as supporting direct reading center de-identification of the data and transfer for use by the heart failure AMP, but also broadly by the scientific research community. We're also actively exploring options for image data analysis to be able to more robustly support machine learning in the ecosystem. We're providing access in terms of the governance that's aligned with the patient consent from all the retrospective studies that are being used for the program as well. We're continuously developing HeartShare and the Heart Failure AMP have been a driver project for Biodata Catalyst, and so we have been working really to create more robust infrastructure to support the imaging needs of the heart failure AMP. But we are meeting the needs of the broad heart, lung, blood, and sleep research community as well. We're working with ODSS to implement digital object identifiers through DataCite, to be able to provide provenance and metadata that can follow the data that's being accessed. We're also expanding support for new data types as well, including single-cell omics, cell characterization, and proteomics. So it takes a village, and there's dozens of folks that have contributed to the success and development of Biodata Catalyst, and wanted to thank them all, some of which are in the room today, and really appreciate the time for everyone today. Thank you.
Thank you so much, Sweta. It's lovely. I'd like to introduce Matt Bookman. Matt is a longtime software engineer who pivoted his career to studying computational biology at Stanford and spent the last 10 years as a solution architect on Google Cloud and Terra, and now the Verily Workbench. Matt received his master's degree in computer science from Stanford and a BS in applied math from UCLA. Go, Matt.
Great. Thank you. So yesterday I got to talk about the breadth of tools available for PD researchers for AMP-PD, and today I want to go a little bit deeper into the researcher workbench and the researcher experience for that. The timing for starting the AMP-PD project, we started at the beginning of 2018, and it followed on to this joint blog post that people from Vanderbilt, Verily, University of Chicago, Santa Cruz, and the Broad Institute had put out, which talked about sort of a new vision for ways for researchers to come together, and the idea that was expressed was, the traditional approach was to make data available and allow researchers to take out the data, which was a great step. But that led to a number of issues that they wanted to solve. Some of the issues was now you have lots of copies of data all over the world, which decreases the security of the data. You also then have the cost to the community. You've got to host this data. As the data gets larger and larger, and we start talking about hundreds of terabytes of data or petabytes of data, that's a whole lot of storage around the world that people are paying for. And then you end up with these silos of not just of data, but of tools and results. And so the idea was to create a researcher workbench, or in the cloud. You now have these public clouds, AWS and Google Cloud, Azure, that are emerging that provide compute and storage resources. So the idea was, let's take advantage of those and create a place to bring researchers and their tools to the data rather than the other way around. And there was one point that I wanted to add, which was really highlighted by Mary Macarius yesterday when she was talking about the GP2 program and their discoveries in terms of Parkinson's and for people of African descent, was this idea that they were in the process of onboarding researchers who aren't necessarily associated with elite institutions. They don't have large compute clusters available to them, but they're able to, essentially with funding, a credit card. In this case, I think the Michael J. Fox Foundation is actually funding the research. They have an entire data center, some of the premier data centers in the world available to them, which they would not otherwise have. So in the case of the Terra platform and Google Cloud, which were used for AMP-PD, some of the advantages to the program is rather than needing to fund a whole lot of tool development, we could take advantage of the fact that there are already thousands of developers, both working for Google as well as in the open source community and the research community, building out tools that are constantly improving the platform. The Broad and Verily were working together to build the Terra platform. So you have a team of researchers adding new features. You have programs associated with the Terra program, such as the Anvil program, that brought community contributions to the platform. And on an ongoing basis, you have users of the platform who are adding new tools, new workspaces. So for AMP-PD, we took advantage of this, and I'm going to go through a few examples of how this worked for us and then how it impacts the researchers. So as a team, so the Verily team, we worked with the Technome team to run a lot of workflows to do data processing. We ran a lot of Jupyter notebooks to do quality control and create visualizations so that the data that was being produced for AMP-PD was high quality. And we did that on the platform itself, and collaboration is built into the platform. And so we can immediately, when we're done and we're releasing data, release the very same code that we ran and make it available to researchers. So if you're doing RNA-seq data and analysis, and you want to bring your own data and you want to run the same workflows, you can do that. We've made those available in a workspace. If you'd like to look at the data and the quality control that we ran, you can run those notebooks as well. And that also provides the starting ground so that you know how to work with the data, and you can launch your research from that. I mentioned this idea of not needing to download the data, and and that can be a really big burden for people from all sorts of organizations to fund a place, to get your IT team to help you set up the compute infrastructure. You might even need to ask them to download the data, and then you're going to, let them know, by the way, I'm going to do some processing, and that's going to take, that's going to take up the entire cluster that we share with one of the other departments. And in the new approach, and I did try this the other day. Literally, it took me 5 minutes to go from the idea of I now have access to the data, I now have my own workspace, and I have a notebook running with some of the data loaded, and I can scale up my computation from there. So this is just a quick view of what that looks like. We make a whole set of workspaces available to researchers to help them get started. It also is important for the platform, as I mentioned, allows for collaboration. And this is another sort of conflict that people can run into when trying to collaborate across organizations is, so I'm able to run my own workflows or my analysis. All of the data is on my institutional cluster. How do I, across another organization, get you access to my tools and data? I either need to copy it or work with my IT team, and sometimes, that just doesn't happen. And so this is another example of how this impacted us as a program, and is extended out to the broader community. We had been running workflows, doing a whole lot of transcriptomic analysis, producing analysis-ready data, and needed to know if we were if all of the samples had been properly labeled before we ran, the next set of workflows, and we needed to run some sex checks against the clinical data. And just in brief, this shows, we had a workspace in Terra that this spans, what did I say here? This is 4 different professions, 3 different institutions, and within just a couple of days, we were able to work in the same environment to produce what truly were to us these lovely graphs that showed that the samples that we were processing did indeed have the right labels as we compared the RNA-seq data to the clinical data. I will close my time with just a look ahead. It's been raised very often for the next stage of AMP-PD. It's been raised over the last few years. We'd really love to enable more joint analysis with AMP-AD in particular. And so there's a lot of opportunities for that, and there's great questions that we want to figure out, and I think we'll talk a little bit about this in the discussion that follows, which is what are the right ways to make this happen? Does it involve aggregating all of the data under one platform? Does it entail improving the federated nature and the ability to interoperate between the platforms and with the data? What should we do with regards to harmonization of the data itself? Thank you.
Thank you so much, Matt. Takes a village. Our last speaker is Anna Greenwood. Anna is the director at Sage Bionetworks, where she focuses on advancing dementia and age-related team-based research and an open scientific approach, and she oversees dissemination of data and results through community data portals. So with that, Anna, thank you so much.
Great. Okay, so today I'm going to step outside of my AMP different AMP programs to help provide an overview of the goals of the SBI program, which is a forward-looking AMP to combine data. And so you've heard about this the past few days, but I just wanted to kind of start with some high-level goals of the AMP SBI initiative. So many of the diseases studied in the AMP programs are characterized by inflammatory processes. And so the goal of AMP SBI is to identify both the shared and distinct mechanisms that are happening in subsets of patients across these multiple diseases to get a better understanding of potential treatments. And so this project seeks to leverage the breadth of the existing data sets that you've heard about for the past day and a half by integrating data across these projects. So what does this mean functionally? And we have all been part of a few different working groups to start to build out a vision for how we might start to enable data integration across these platforms. And really, when we think about integration of data, it's these 3 main pillars that we need to think about. So one is the data itself. So data is coming from patients, from different sorts of tissues, from different environments, a brain bank, clinical sites. It is processed in different ways. It is sometimes from historic samples, sometimes prospectively being collected. And all of that influences the harmonizability of that data and even the potential for that going forward. The infrastructure itself, we've heard, Google Cloud, Amazon Web Services, some local servers might be used for some of the data. How do we make those systems compatible with each other? This is technical problems that have been solved by many tech platforms. Amazon, Google, they've solved this in many different ways. But how do we take those best practices and think about how to build towards a future? And then finally, governance, right? We have to make the data as open as possible while respecting the patient consent and patient privacy. And that is interpreted in very many different ways across these AMP programs. And underlying all of this is not just, the technical work, but it's the people work. This is integrating data. Infrastructure requires not only the the tech. We can't outsource this to ChatGPT, despite what we may want to do. We're going to have to get in there and do a lot of this work. And so, just summarizing all of this, some of these slides come from some of the working group materials. But the types of data and sources and tissues sampled really differ across these AMP projects. And we haven't even included some of the forward-looking projects here either. The data repositories themselves differ across AMP projects. And so these are just the front-facing web portals, but the underlying infrastructure for these is all quite different as well. And then, of course, the governance and data access policies differ across AMP projects. And one thing that I think is sort of lying under the surface here that we haven't explicitly brought forward is that there are varied end users and use cases that are being served by these different AMP projects, right? So computational biologists, many of the academic researchers, many of the pharma partners are computational biologists. They want the raw data. They want to get in there, reprocess it from the ground up. But then we have people, clinicians, patients, patient advocates, experimental biologists who may have, like me, many years ago been able to use R, but now have completely forgotten that. And so, I don't want to dig into the raw data. I want a more summarized view. But really, and so, with that lens, the potential for the reusability of the data really varies by data and user type, right? So raw data is arguably the most reusable because you can start with the raw data and you can do almost anything to it. And this is true, I've laid out here for gene expression and genetic variants, but this is also true for imaging and other kinds of data modalities. Analytical results, we've made a lot of decisions to generate analytical results like differential gene expression in 80 cases versus controls. How did we define an 80 case? Maybe you don't agree with that, right? And so you want to rewind somewhere earlier in the process and start fresh. And so we find generally that the process data, this middle layer, is probably the most highly valuable to the broadest community, but there are obviously nuances depending on which user you're talking with. And so the different AMP programs have tried to develop solutions for these different data and user types. So just an example from the AD Knowledge Portal, you can access raw process data, analytical results that are paired with publications, and you can also access our curated results explorer, Agora, in which you can pick a gene and you can look at differential expression of that gene. You can look at differential abundance of the protein in brains of Alzheimer's cases. And the other AMPs, sorry, this slide didn't render quite as, oh, it's better up there. The different AMPs have different, we have portals, we have explorers, we use sort of different terms for these, but we're all aspiring to kind of reach a different spectrum of users in different ways with our products. And so, okay, with this landscape, how do we move forward? What do we do going forward? And we all hear the term data silos. I think this is a little bit of a pejorative term. These are purpose-built silos, right? We built them to serve the community. Like all of these AMPs, we worked with stakeholders, we got into a room, we designed the system. And now, of course, in retrospect, yes, they're a silo. It makes it harder to look across. But we can, let's move forward and think about what we do to move forward. So the first, from a user perspective, we want to easily find compatible data and results from different systems. We want to be able to seamlessly access that. And then we want to be able to potentially analyze that data in a common environment, which you heard in a nice way from Matt. And so to start building out and prioritizing what we're going to do in this new initiative, there were some pilot projects that were funded to start looking across AMPs and disease areas and see what issues and challenges these teams ran into. And so a range of projects were funded, including evaluating shared inflammatory signatures across diseases, looking at shared genetic association across diseases and immune traits, and integrating single-cell or single-nucleus transcriptomic data from tissue-derived immune cells. And one of the requirements of these projects was that they had to include not just neurodegenerative diseases, but both neurodegenerative and other systemic diseases. And this is just a really quick example. So these projects have been funded for about 1.5 years, close to 2 years now. They're starting to generate results. And this is a nice example of a network analysis that was done from transcriptomic data from AD brains, and then also from type 2 diabetes, so adipose tissue in this case. And you can see that some of these hub genes in AD are actually similar to what's being found in type 2 diabetes. And this, if I didn't say it, is from Bin Zhang's group at Mount Sinai. And so this is, sort of shows one of the promising features of what we're going to be finding when we combine across these diseases. And so this now brings us to the next phase of data interoperability for the AMP projects. And this is what's being called as AMP Adapt. So the focus of AMP Adapt is to create this federated interoperable data ecosystem so that people can probe multi-omics data across multiple diseases. And now there are working groups that are getting together to develop the project aims. One is really focused around data and platform interoperability. The other is data governance. And then finally, that will enable analysis of shared and distinct pathways of inflammation across disease. And I think, Susan, did you want to?
Well, unfortunately, Courtney has let us know that we need to move to the next session, but I do want to thank each and every one of our speakers. This is a great vision. I look forward to moving this program forward with collaboration from our colleagues sitting here and across the audience. So thank you for your attention today, and hopefully we can connect offline.